So anyway, for today, just want to discuss a bit why ticks and tick-borne disease might be affected by weather, climate, and then climate change. And to do that, because you probably could read some review articles and know about that, I really want to talk a little bit more about the biology and the ecology in particular, such that you remember what is it about them that makes them kind of affected, or maybe not so affected, especially if you think about compared with mosquitoes or such, by changes in weather and then long-term patterns of climate. And I'll just provide a little bit of evidence, and then also just to remind us that even though the topic is about climate change, there are probably other climate independent factors, as well as some other factors that might be interacting with climate that could be just as important or more important for the emergence of ticks and tick-borne disease. But today we'll kind of dig down into learning more about the biology of ticks, so that if nothing else, you know more about that, and how other factors, such as you might hear about biodiversity or landscape, land use, change in patterns, how those might also affect the emergence of ticks and tick-borne disease. All right, so I'm going to focus on Ixodes scapularis in part because it's very important as a human and companion animal disease, more for humans really, but it affects a lot of people are very concerned about that for their dogs and such. So it's important public health wise, and because of that, it's been very well studied amongst ticks and tick-borne diseases. So because of that, even though there are many other ticks that are important, some of those have been less well studied. And so perhaps what we have learned from Ixodes scapularis, we can think about when we're thinking about these other ticks, all right, and their pathogens. And then partially it's because we have a little data to share. And yeah, I will talk a little bit about some other, just a tiny bit, I guess, about other systems. But I wanted to point out at least two papers that people may be interested. There's a lot of papers now about climate and ticks and tick-borne disease, but here are two that were actually written a few years ago, 2016, that I think break down a lot of the things that of what I'll talk about. But certainly this is also something that's happening in Europe, so there's definitely a literature there as well. Okay, I want to also acknowledge that a lot of my understanding about especially Ixodes scapularis, but about ticks, has really come from working with these folks, where we had a project for about 5-6 years kind of looking at the variation in the ecology of the black-legged tick from the north to the south. And in part it was to help us really learn more about the ecology of the tick in the south. But in doing so, we learned also that ticks, it's not just a north-south thing, but an east-west thing, that they have some differences. And then I want to also highlight that Dr. Genevieve Peng, she had finished her dissertation with me last year, but I'm highlighting just a little bit of her work in this talk. There's a lot more, but she was very interested in how abiotic conditions affect black-legged tick survival and development rates in the field, to understand a bit about the geographic variation of Lyme disease, and also to think about and this is generally the context of a lot of the work that we do, but as northern black-legged ticks move south into a warmer climate, as well as climates are warming, and so that the north becomes like the south, how might we predict changes in Lyme disease and other diseases, excuse me, caused by pathogens vectored by Ixodes scapularis. This is not COVID-19, sorry. All right, so stepping back out a little bit, tick-borne diseases, as some of you might know, are a growing problem. They've probably been underestimated a bit in the past, not just here in the United States and North America, but probably all around the world. And in part, that's because many of these symptoms for these diseases are general kind of flu, fever type of presentation, except for potentially something like Rocky Mountain spotted fever. So, some of these diseases, especially in other parts of the world, probably do get mistaken for, a number of other kinds of diseases. But it seems to be recognized that the importance of ticks and tick-borne diseases for human health, companion animal health, and also for livestock in other areas has really been increasing. And actually, even maybe for livestock in the US, now that we have the Asian longhorn tick that's been invading. So each of these dots represent a case of disease reported to the CDC by the county of residence. And so you see about 6 different diseases being shown here. These 6 ticks are some of the, more frequent human biting ticks. And this panel comes from the same publication as the previous figure. And I would say in most of our areas, up, in the more northern areas, it really is the black-legged tick, the lone star tick, and the dog tick that seem to be more of a problem. But certainly, and especially perhaps further south you are, like in Missouri and such, perhaps maybe the Gulf Coast tick is coming in. But certainly you're inundated with the lone star tick. Okay, let's see. One of the things to note is that these are all 3 host ticks that we call hard ticks, and each life stage uses a different host. And that's important for when we think about why climate and climate change could be important for these ticks and the pathogens they transmit. And so I'll illustrate this by talking about the black-legged tick, or some people call it the deer tick, and go through its biology. So to remind you, this tick transmits several pathogens causing several diseases. And I think as we study more, we'll probably find other viruses and such. But there are 3 post-egg life stages: the larva, which has 6 legs, the nymph and the adults, which have 8 legs, and only in the adult stage do you have the male and the female sexually differentiated. Each life stage requires a blood meal, except for the adult male here. And that is for Ixodes ticks. The Ixodes tick, the male, may try to take a little kind of a sip of fluid from its host, and I think it's for so that it doesn't dry out. But the other hard ticks, like the lone star tick, the dog tick, those males actually do feed a little bit more because it stimulates their formation of sperm. But the Ixodes ticks don't need that. Okay, so here the cartoon of the life cycle that Jenna made. So the eggs are laid in one batch from a mom, and they hatch into larvae. Then the larvae feed on a host that comes by, all right? So these larvae don't move very far. They're ambush parasites. And so they take their blood meal from the host, and a larval Ixodes scapularis tick probably about 3 to 5 days of feeding, depending on potentially what kind of host it is. And then it detaches, and it's an engorged tick, and it falls into the leaf litter, and then it will molt into a flat nymph. Then that nymph will also then attempt to find a host, waiting for some host to come by and pick it up, and it will feed. It will also take about 4 to 6 or so days to feed to repletion, and then it will drop off into the leaf litter, and it'll take some time to molt. And then when it molts, it'll molt into an adult, and here we have the female shown. It could be a male, but we have the female shown, and then the female then looks for a host to feed on, and in this case, we have this poor little Bambi. So, and then it feeds to repletion, drops off, and then lays its eggs, and then she dies. So these ticks as opposed to a flying insect like a mosquito, will not really go too far looking for a host. The adult stage may move a little further, but certainly they just sit and wait for a host to come. And in terms of for the adult stage, really, they hone in on a larger host, and that in part helps them find their mate, such that they're not trying to find their mate out in the, the whole woods or such. So they kind of track to the definitive host. All right. And in northern latitudes, it takes at least 2 years for the life cycle to be completed. All right. So I'll just show you a few pictures now. And most of these beautiful pictures are courtesy of Dr. Graham Hickling, with whom we've worked many years. So this is a clutch of larvae of Ixodes scapularis, and they are questing for a host on a leaf litter, and you can see them raise their front pair of legs. So ticks, as you know, don't have antennae. And so, but what they do have are these Haller's organs, and they're at the tips of the front, the most anterior set of legs at the base of the claws. And they have these sensory organs called Haller's organs that can then, process the chemicals and I guess heat and other sorts of things that are in the environment that can tell them potentially a host is coming by, or that when they get on the host, they can then kind of probe to say, does this seem like the right host, before they then commit and put their mouth parts in to feed. And so again, they don't really move very far from this cluster, maybe a little bit, but then when a host comes by, they'll latch on with their tiny little claws. And then so here are a nymph on the left and an adult female on the right, also assuming the questing posture with its front pairs out, testing the winds, hoping for a host, probably the person who is taking the photographs. This case on the right is Gabe Hamer, who's now at Texas A&M. And again, the larvae and nymphs have a very wide host preference, and I think I have some slides here to show you. And they feed on small and large hosts, whereas the adults are limited more to the medium and large hosts. Yeah, so here we go. Here are some pictures. And so you can see there are mammals on the left. All right, and then you get birds, and there are also lizards as well, especially as we go south. And that's partially because we just don't have as many lizards up north. But even when we do have lizards up north, these black-legged tick larvae and nymphs will feed on them. More so the skinks, which is the lower lizard on the right, than the fence lizard. But if you know about Ixodes pacificus, the western fence lizard, you know that the western black-legged tick, the western fence lizard is a favorite host for the western black-legged tick, and it's just covered and nailed by them in the areas where western black-legged ticks are very abundant. And similarly, yes?
I just have a quick question. I know in Western California that the lizards have a borreliocidal factor, so it actually clears them of the Lyme infection. Is that true where you're at as well?
Yeah, so these skinks, Michael Levin showed in a laboratory experiment that they can be infected and they can be reservoirs, but in the field, we have, by assaying the larvae, which we pull off from the skinks, we have never found them to be infected. And then a graduate student of Graham's tried to do a reservoir competence experiment, and she also did not seem to find much transmission. So even though Michael Levin did find transmission, don't know if that's because, well, maybe in his hands he was able to do it a little better, or there could be strain variation in the Borrelia burgdorferi, that sensu stricto that we used. But certainly from all the hundreds of ticks that not just my lab, but others have looked at off of skinks, we haven't found any infected. And so if there's some infection in nature, could be not very much, probably not ecologically relevant. But yes, I think they do also have this borreliocidal complement, which is what it is in the Western fence lizard. And out in California, they had also looked at the alligator lizard and at least one more other lizard, and it was it is a complement, so part of the innate immune system that's able to kill the Borrelia. And what's interesting is then if you put an infected a nymph on a skink or a fence lizard, after it feeds to repletion, when you take it off, it's uninfected. So does that answer your question or comment about it enough?
Oh yeah, that's great. Thank you.
Okay. Yeah, and that is the same mechanism for how for why the white-tailed deer is also not a reservoir for the Lyme disease pathogen. All right. And so here are just a range of some more medium-sized and large hosts. And there's, others like skunks and possums that I didn't put in here, that it seems like they range in their ability to be reservoirs for Borrelia burgdorferi, from weak to moderate. The rabbit is a pretty good host for Borrelia burgdorferi. And since we're on this topic, I guess I'll just go back and say most of these small mammals are pretty good hosts for and reservoirs for Borrelia burgdorferi. Vole, which is this the one where I'm showing where we're holding it with a purple glove. That seems to kind of vary these different voles for their competence. But the shrews in the left, they're very good reservoirs. The chipmunk and the mouse, very good reservoirs. Over here in the birds, there's variation, just like you'd expect, just like we see amongst mammals, amongst birds. So robins, thrush families are very good, but this catbird is not a very good reservoir. And like I said here, for the most part, lizards tend not to be reservoirs. Are very weak. And I don't know of any reservoir competence study, but a coyote here on the bottom left, I would think that they would, overall, they should be, I think in theory, they should be fine as reservoirs, because we know that dogs can be infected. And there are a few papers out there, I think, that have shown that, you can get infected ticks off of the coyote. However, I don't know, for instance, how long a coyote can be infected for. Is it like a mouse, infected for life? I'm not sure. Squirrels aren't supposed to be that great of reservoirs, and neither are raccoons. Not sure about bear, but certainly the white-tailed deer is not a reservoir. And so that can be confusing for a lot of people, especially because it's called the deer tick, and because we often tell the public that a large reason, an important reason why Lyme disease has spread so easily, I guess, or so quickly, is that in part, in many places, that once the deer populations got to be, it's a very low bar, I think more than like 9 per square mile or something. But as our deer populations in many places, due to the management, to the, one would say the excellent management of the Fisheries and Wildlife departments that have agencies that have brought, they brought the deer back from near extinction in the 30s, 40s, 50s, 60s, but then they've run rampant, and they are in our neighborhoods, areas that, because of that, and because of their ability then to feed lots of ticks and maintain the tick populations, that we interact with them a lot. Right, so it's not just that they're out in the woods and the ticks are left out in the woods, but because we have modified our habitat to bring deer into our neighborhoods, that means that they can bring the ticks as well. All right. Okay, so getting to climate change and such, and I put in this dog tick just to have a little diversity of ticks, right? So why are these ticks so, could be affected by weather, climate, and therefore climate change? And it's because, even though we think of ticks maybe because of our experience or because we have dogs and such, but ticks spend 99% of their lifetime in the environment. We see them for maybe the few days that they're actually parasitic. And so because of that, they are really affected by the environment, because they, first of all, they don't have any alternative energy source, all right? They are obligate feeders. So when they are out in the environment and I mentioned how, at least for the black-legged tick, it has at least a 2-year life cycle. So if 99% of that time it's in the environment and it's just living off of whatever energy sources it received in the previous blood meal, or if you were a larva, what was in the egg. And so they are living off of that, and off of that, the fat resources there, and their survivorship when they're in the environment like that depends on them being able to survive long enough, to find a host, right? And they are ectotherms. And so during that time, they are affected by the temperature, right? And they're very prone to desiccation. And those are, the desiccation part is probably like the kryptonite of ticks, right? So there are things that they can do. I mean, they are actually a very successful group. They potentially have been around since the Devonian. And so they've been able to survive different ranges of climate, but they have behavioral adaptations. And then there's also diapause and other life history adaptations that probably have helped them try to overcome some of these challenges due to their biology. Having said that, they have tons of babies, right? So part of this is just a strategy, and our selected strategy to just have a lot of offspring and hope that at least 2 make it. All right. So talking about how ticks might be affected by temperature, and RH I have for relative humidity. So, and these factors are picked because the ticks are ectotherms, and then they're prone to desiccation. So in terms of temperature, like many organisms, there's some optimum for their survivorship, and that's a little different for larvae, nymphs, and adults. But for the most part, and these are mostly temperate ticks, they can survive. They can survive so much. They have some cold hardiness. And actually, some of that research was done by someone, I don't know if anyone knew, but Van Dyke from Iowa and others. And so they can survive temperatures down to minus, I think they looked at like minus 12, minus 20 C. They can't survive like that at those temperatures forever, but they have some tolerance there. But what's important to know is that these experiments, and we've done them in the lab here too, they kind of tell you what theoretically their limits are. But in nature, these ticks aren't just, they're not just sitting on top of the leaf litter and being exposed to the elements. They are down in the leaf litter, in the duff layers, where they're buffered from some of the colder temperatures above and drier temperatures above. And so, for instance, also snow cover provides a good buffer. So in Michigan, the ticks are really, in lower Michigan, the peninsula, they kind of took off around Lake Michigan, near up the coast. And part of it, and that's the area where you have the apple orchards, the peach orchards, the cherries, the grape vineyards and such. And that is in part because the lake moderates the climate, but also they have the lake effect snow. And so I think that really helps buffer a lot of the organisms that live down, that could be prone to drying out and being cold, but they have little snow blankets over them to help them pass the winter. At the other end, what happens is that when the temperature gets really high, I'm sure like many organisms, all of a sudden the enzymes and such, they don't do as well. And then also, they're going to be burning through their fat reserves a lot more. But when it gets really high, what happens is that the integument then, the tick will start to get damaged, and then you'll have water loss. So it really comes down to water loss. And so ticks then, to try to avoid water loss, there's 2 things they can do. Well, they're related. They go down into leaf litter where it's more humid. First of all, it's just more humid. And then also they can exude some secretion, it's a little salt, you know, on their mouth parts, and then water vapor around it can condense, and then they can suck that back in. So that's I think that's a great, you know, secret weapon that they have there. And I wanted, Graham had this nice slide that I wanted to share with you guys. But these are data taken from Knoxville, Tennessee in April. There's no wind and there had been no rain, all right? And 75. So it's kind of in the shade. That, you know, in general, that's really, it feels like, you know, when I'm going out to collect for ticks, that's pretty good, you know, pretty ideal actually. So, but the reported relative humidity, you know, seems kind of dry and actually when we go do our sampling, oftentimes we try to say to our students that if it's not 50% or above, let's not go because it might be too dry, all right? However, there are, you know, it really does depend on the environment a bit. And what you can see here is on to the left of that plant, on the x-axis, we have humidity going from lower to higher. And then on the y-axis, we have kind of the height above the leaf litter, and it's to correspond with this picture of this plant. And so, for instance, that's 10 centimeters above the leaf litter, and there you have relative humidity at 44%. So the reported relative humidity of 40%, actually, I'm not sure if he got that from a weather app or if it's from actually what you would call it, a data logger that may have been, we often have had data loggers at 1 meter above the leaf litter. But here you have 44% at 10 centimeters above, and then at the surface, 49%. And as you go lower, it gets much more humid. And down there, see, I think, yep, down there, 80%, that's still not optimal for these ticks, but you can see how it's getting much higher. All right, and so ticks, they climb up and down, they climb up and down the substrate of the leaf litter as a strategy to find a host. But as you can tell, when they are higher up, the relative humidity is lower, and then so they could be losing, you know, their water balance is off, and so they balance that by having to go back down into the leaf litter. Now, I'll tell you that this is something I'm not sure actually if it's the right way of thinking, but the paradigm in the past was the story I told you, and that the emphasis was that the ticks would find hosts that, when they come up above the leaf litter. And so there's definitely this interplay, this trade-off of being above where it's not humid, to then having to rest, go back down into leaf litter to rehydrate, but yet have the trade-off of not being able to find a host. I'm not really sure if that's true, and in part, I think it's because there are plenty of hosts that could be below the leaf litter. And so in the north and in the south, you know, there are shrews that really travel a lot under the leaf litter. And in the south, the skinks are really traveling a lot under the leaf litter. And so, and they are the hosts that are nailed by these ticks. People don't really know how the ticks get on the skinks or how they get on the shrews, but it could just be that hanging down under the leaf litter might be good physiologically, and it may not put them at a worse, at a disadvantage for finding a host. It's just that potentially coming above that gives you other alternatives, other hosts. And maybe if those other hosts are more abundant, then perhaps that is the strategy. All right. But anyway, if you read about ticks and in the literature, then you'll see that paradigm of that trade-off, that you only can find a host, you know, above the leaf litter, and that's the, you know, riskiest place. So, all right. Okay. So Katie Berger, as a graduate student in Rhode Island, she did this research project that showed that when the relative humidity is really low, that's when ticks are more prone to desiccate and die. So she called these tick adverse moisture events, TAMEs. And what you see is on the left graph, you see that the total number of TAMEs, which are 8-hour periods where you have the relative humidity in the litter be less than 82%. So what happens that you see as you increase in your number of TAMEs, then the total abundance, they had dragged for black-legged tick nymphs, total abundance decreases. And then to try to associate that really with mortality, because you don't know, there could be other factors going on there. She has the right graph, which is they had visited each site twice. The first time, sometime earlier in the nymphal season, or peak nymphal season sometime maybe, and then another time still when the nymphs should be active, but later. And so then you have the same x-axis, the total number of tick adverse moisture events on the x-axis, and on the y-axis it's the ratio of how many ticks you found the first time when you went, which is the numerator, to then the number of ticks that you found the second time, which is the denominator. And so if the ticks were the same in both events, your ratio should be 1. But given that there's always mortality, you should see that the numerator is larger than the denominator. But here, certainly, what you see is this positive slope. And so we don't know for sure if this is just due to TAMEs, but certainly this, because ticks may leave also due to finding hosts, but certainly this is correlated with the number of adverse events here, not just with time. Okay. All right. Howie Ginsberg, also at Rhode Island, one of our colleagues, he then also did this experiment. In my group, we raised lots of ticks from different areas of the U.S. or Eastern U.S. And we had, because we were interested in their different ecologies. And so we gave him some of these ticks that we had made. And he has 2 publications for it for 2014 and 2017. But these are laboratory experiments. And basically what you see, and this is for larvae, is that in general, when you look at how long ticks will survive for in incubators, in these chambers, you see that they decrease over time. All right, you'd expect that. But the decline of that, though, is increased, or the slope, the severity, as you decrease the relative humidity. And so this is just to show how, again, humidity is important for survivorship, but that the N stands for how the ticks were in chambers that had northern conditions, which were taken from, I think, Rhode Island or Cape Cod, the conditions we had in the field there. So a certain average temperature for the day and average temperature at nighttime that was cycled. And then the conditions for the South were from, I think they were from Florida, our site in Florida. And so again, so they would be hotter, but they were the relative, and the different relative humidities. So it seems like for the black-legged tick that the northern conditions are more suitable for their survivorship, and the larval stage is the most susceptible compared to the nymphs and the adults. Okay, so Nick Ogden, through field studies with Robin Lindsay, had seen that it's kind of interesting to think or believe, but there's a constant daily per capita mortality rate. And so if that mortality rate is constant for the tick in its lifetime, then the longer a life cycle is, actually the chances that it will survive are lower. And that will have effects at the population level. And so, so all things, equal, warmer temperatures should speed up. And sorry, I think I've reversed the slides here, the mechanism, but warmer temperatures should increase the rate at which each life stage develops, and therefore lead to a shorter life cycle, which means a higher survivorship and then perhaps larger population sizes. And this is obviously until you run up, until you get to temperatures that are therefore become detrimental to the tick. All right. So this is really kind of operating in the area of the temperatures that would be more beneficial than negative to the tick. And if you're wondering about, well, how is there this constant per capita daily mortality rate? I think it's in part due to the fact that the ticks can, that they are picking their microhabitats. And so there's refugia in the leaf litter. And then also things that I haven't discussed yet, but there can be, they can become quiescent. So they decide not to be very active at all, even though the temperatures and humidities are such that they could go out, but they say, I'm not going to, I'm going to stay down in the leaf litter because it's better. And there can be diapause, which I'll explain in a bit, which is a genetic programming that's due to evolution over time to try to avoid bad conditions. Here, yeah, let me see for a second. Sorry, I wanted to show. I think this was supposed to be earlier. This is how temperature, how it affects, temperature effects on development. And so as on the, this is from Ogden et al., 2004. On the x-axis, you'll have these are laboratory experiments, but you have increasing temperature. And then on the y-axis, you have the days to, and this is for development, for the different stages. So in the upper left, it is about how quickly will an engorged female lay her eggs, right? So what you see is that at lower temperatures, it takes them a long time, maybe 200 days to oviposit, all right? But as the temperature goes towards 30 degrees Celsius, the number of days drops much lower, probably less than 20 or so. And you see the same pattern then when you look at, in the next slide down, once you lay the eggs, the eggs then will hatch into larvae. And so you can see at a lower temperature, it takes longer for them to hatch. And then a higher temperature, it takes less time. And you see the same for then the larvae, engorged larvae to nymphs, the upper right panel, and then from engorged nymphs to become adults in the bottom right panel. And so this, then relates to this idea then about how temperature may affect not just physically, like with the damaging of the integument or freezing, how it could have impacts on mortality, but that at a life cycle level, when you consider also development rates, how a temperature can affect the population of a tick. And here is just showing you Troughton and Levin raised ticks in the lab. Lots of people have colonies of ticks. And this is in the situation where they're raising them at 22 centigrade. You have Ixodes scapularis at the top, and there's several different other ticks here. And, they can push a life cycle through, a generation of ticks in less than a year, 160 days or so. All right. But in the field, these life cycles tend to be longer than that. And in part, it's due to temperature-dependent development. And then other factors, such as, in the lab, once they molt, they wait probably a week or two for the ticks to harden, and then they feed them on a host right away. The ticks don't have to worry about how much longer it'll take to find a host. In the field, that's not always the case, obviously. And so that could, so density of hosts as well as seasonality of the hosts together may affect that. And then again, the environmental conditions in general. And I'll talk about diapause. All right. And this is my little joke or pun. Diapause adds pauses to the life cycle. So in case some of you have not heard of this term before, I'll go back to say quiescence is just when the environmental conditions aren't so good, the organism decides to behave in their best interest. So for instance, if it's really cold outside, we just don't go outside, many of us. Or if it's raining, we don't necessarily go outside. We we're adjusting to what the environment is, how it is. But diapause is, even if the environment is good, it's fine, it's sunny, it's warm, you want to go out, genetically, there would be a signal that those cues don't trigger your ability to go do whatever you want. And so this is how ticks have this genetic programming, and other organisms too, that actually are often cued by photoperiod. So the length of a day, getting longer or getting shorter, which is representative of the seasons, that over evolutionary time has been selected for to help reduce mortality. So examples of developmental diapause. So winter might be an unfavorable time, cold and freezing and all that. So, out in Iowa, just like here, your females that are out in the fall, they will engorge, and if you bring them to the lab, you can get them to lay their eggs. All right. But out in the field and in the lab generally, if you don't, if you don't observe, I guess, the photoperiod. But in the field, those females, they don't lay their eggs until the following spring. Now, some of that might be the temperatures are cooler, but we know that from those experiments that Nick, had done, that actually some of the temperatures, in October, November, they're fine. The females could lay her eggs then, but they just don't. So females that are fed in the fall and then in the spring, they all lay their eggs in the late spring. All right, so that's an example of developmental diapause. And similarly, larvae that feed late in the summer, they don't molt then into nymphs until the following spring. Whereas the larvae that feed earlier, they'll molt into nymphs, and then as the nymphs, they'll just be flat. They'll just wait. All right. Ixodes pacificus is something different. It's not the winter coldness, but summers in the areas in California where these ticks are found can be a bit too dry. And so in that case, they're not active then. But you know what? I made a mistake. I bet that should be under behavioral diapause, which is the next category. So the behavioral diapause is we're not talking about development. The tick is already in the molted stage, but yet they don't behave they don't behave as they could to find a host and such. So nymphs that were, that just molted from larvae that before the fall, even though there are hosts around that they could get on to feed, they don't do that. They wait till the next spring. And similarly, when the nymphs do feed in the next spring, summer, they will molt into adults. All right. But those adults in June, July, August, they're not host seeking. All right. They will wait till the fall. And so that's called a behavioral diapause. So these are triggers that aren't affected, they're affected by day length, right? Which won't change, due to climate change. All right, they're on a totally different cycle. However, in the lab, certainly, we can override some of these cues. And so then the question is, with climate change, will some of and when we override them in the lab, it's often because temperature is overriding them. So that's a question in the future, will climate change will some of these warmer temperatures override some of these cues, or are these, or will it not? But I think in mosquitoes that can happen. All right, so let me skip that. I know it's kind of we're getting to 5:48, and I think I'm only through some of the slides, but let me just, let me do a few more and then we can decide what to do. But the temperature and relative humidity can also then affect, when the ticks are active. So diapause, as I illustrated before, will affect then when ticks seasonally might start host seeking. But temperature effects then within that, okay time for them to host seek may affect when they actually begin host seeking. So for instance, in 2012, it was really warm here in the north, and nymphal ticks were host seeking in February. And so they usually don't host seek until about March or April. But obviously that must have either the temperature overrode the diapause cue, or the diapause cue was set by, let's say, December 21st, the winter solstice. And so it's just that in normal times where the temperature is cool, the nymphs aren't active, even though perhaps their photoperiod cue is passed. But then with temperatures increasing, strongly enough, they were able to be active. Okay. All right. So I will just do the predictions and evidence of climate change. Actually, there's just a few, but a bit it's what you would expect. Ticks are expected to move towards the poles, all right, as climate warms. And that's because all of a sudden now you have areas there where they can survive the summer or longer periods of time, enough time that they physiologically can survive, right? And then also they have time to find a host and potentially molt to the next stage. Equatorial areas, maybe some of those ticks, maybe it might get a little too hot, all right? But some evidence that we have seen ticks expanding due to climate are areas like in Sweden. So this is Northern Europe and Sweden. They've seen ticks in the areas move from the coastal areas, which are warmer, in the 80s, then further inland where it would have been colder. And similarly in Europe, they've seen ticks in Switzerland, in I think the Czech Republic, move further up mountains where they hadn't been before. And in the US, the Appalachian Mountains, although they're not super high, tall mountains, similarly in the past, they did not have ticks up at the, near the top, but I think they, Graham Hickling and others have started to see more black-legged ticks going further up the mountain. And of course, Nick Ogden, Canada, the ticks have been spreading into Canada, and some of that is due to just the numerical spread, the abundance of Ixodes scapularis moving. But we have experimental evidence from before, from Rob and Lindsay, when the ticks could have survived there, but just really, or may not have been able to survive, but over time now, it's warmed up where they can and be able to find hosts. Another, it's not just that temperatures are getting warmer, but there's more precipitation in many places, right? And so I think especially out towards where you are, and Minnesota, Nebraska, and the Dakotas, in the Great Plains, where you've had more grass and less trees, and that kind of habitat that's suitable for Ixodes scapularis, you have seen, I think in Iowa, as well as some of the other states out there, that near rivers where you do have more trees, that's where Ixodes scapularis has been found. But as there increases in precipitation out there, I would assume that then you'd have a little bit more shrubs in many areas, and then perhaps the areas would expand to be able to have habitat for Ixodes scapularis. I'm going to skip this slide, but this was about that point, about how you can have ticks in some prairies, but they have to be really, the prairies are probably a little too dry, but if they get wetter, they could support more. This is just evidence here, about how the growing season in the US has, for the most part, increased over time. And I think that is going to benefit then lots of these ticks. Maybe not so much, and this is very interesting to me, but in the southeast, where actually the growing season has decreased a bit, and that area is supposed to get a little cooler as well from, I think, more rain. I actually think that might be better for Ixodes scapularis, because right now Ixodes scapularis is down in the south. However, there's not much disease, but as the northern ticks move down south, this constellation of the environment with the northern tick might be more like some of these more northern areas eventually, and maybe it'll help them. I don't know if it'll be to that extent, but I think it'll give the northern tick a little bit more of a higher probability of being able to survive and transmit Lyme disease pathogen there, versus as it moves down now and it's very warm. All right, so that's Jenna's work, but do we have, I know it's 5:54, but do we have a few more minutes I could tell you about how pathogen transmission might be affected? Because some of this might be of interest, I think, especially.
Sure, Jean. I think, you know, because this is so much more free form, we aren't trapping people in a lecture.
Okay. That's right. If anyone needs to leave,
Yeah.
I won't be hurt, you know, but I'll just share this part because it's showing you, I mean, it's showing how it's not just about, yeah, pathogens. All right, you've been waiting, Brianna. So, but it's not just about thinking about tick survivorship and, yeah, an abundance and then their ability to move, but it's actually how the transmission cycle may be impacted, all right? So phenology, that is the seasonality, right, of organisms. So I just have here, you know, about the plants and the trees, you know, flowers starting to bud. It's about the birds coming back, you know, and you've probably heard that the birds seem to be coming back earlier each year, right? So here, in terms of transmission, the black-legged tick, when the larva hatches, it hatches free of the infection. So the only way it can get infected by the Lyme disease bacterium or most of the other bacteria and protozoa that it transmits, it has to acquire it from a host, all right? So this is an infected mouse. It feeds on the mouse, it becomes infected, it molts and becomes a nymph. At that point, that's the first life stage that can be infected and then infectious. All right. And that's the life stage most important for infecting humans, by the way. Okay. So the top graph here, what you see for all the graphs, but the top graph is, again, the paradigm, especially from the Northeast, that has been set forward about how this transmission occurs so well, or the maintenance of Borrelia burgdorferi in nature. So you have in pink is the nymphal tick, and when it's active, its relative activity, from March to November, and then you have the larva in the blue curve. And so what you see is that you have nymphs host seeking before larvae, and you have this because you have a 2-year life cycle. So nymphs, the first stage that can be infected, when they feed on hosts in June, those hosts, you know, they feed and feed and feed on the host community. Then when it's later in the summer, when the larvae are coming out, they're able to feed on those hosts and then become infected. And so this, what we call asynchronous asynchronous activity that's also reversed, where you have the nymph before the larva, is what allows transmission or maintenance of Borrelia to occur in an area via the host community. So how would climate then changes, how would they affect then this pattern over time? So you could have, and so these are some options down below, you could have increased asynchrony where you have the nymphs, you know, spaced out a lot from the larvae. You can have synchronous, or you can even have larvae preceding nymphs. Now, this is only important because most of the hosts that are reservoirs, good reservoirs for the Lyme disease bacterium, and I would say Anaplasma, Babesia, and such, they're small mammals or small, shorter-lived organisms. All right, because the one drawback with this, the best, you know, supposedly scenario in the top, is that if a nymph infects a mouse in June, that mouse has to be infectious and living by the time larva comes out to feed on it. And so that gap between nymphs and larvae feeding is really partially, it's amplified by the fact that these small rodents may not live so long. If instead, if we're talking about a deer, let's say it's a deer-borne disease, then it may not matter as much, because although they have mortality too, they're more long-lived. Yeah, and maybe they would even carry the infection if they're, you know, to the next year. But now you can think about then immunology. And so, you know, the white-footed mouse and chipmunk and such, supposedly they're infected for life. And so again, for life means how long it lives. So the worst case scenario for these pathogens, you know, is if the larvae feeds before the nymphs, the bottom right, because if that's what happens, you're not going to have transmission. But for these other scenarios, we still might have transmission. And actually in the Midwest, the northern Midwest, we have a lot of this synchronous transmission. And then you can see here, perhaps, and for short-lived pathogens, that might actually be better, because perhaps the nymph infects a host, and anaplasma, for instance, is in a mouse maybe for 2 weeks. Well, if that's the case, then perhaps that's why, I mean, if you had larvae feeding on these hosts at almost the same time, they're then more likely to get anaplasma, pick up anaplasma there than they may if they were in the Northeast. So we're interested in that. And actually the other aspect to this, and this is just showing that difference, in the Midwest, you have the activity patterns of the two stages overlapping a lot more than the Northeast. And what you also see, I'll skip here, that was lab experiment, is that the strains of Borrelia burgdorferi, although we say the mouse is infected for life, that's for certain strains, and not all strains do that. And so what you see is that the Midwest actually has a greater diversity of Borrelia burgdorferi strains than in the Northeast. And that's because potentially with greater overlap, this kind of phenology might rescue some of these shorter lived strains in these hosts. And so people are interested to know if that's one reason why potentially in the Northeast you have the incidence of Lyme disease is much higher. It could be because the strain that's there that lives longer in mice over time, maybe that is more virulent. That strain makes up only 50%, or actually much less. It's 50% of the strains out there in the Northeast, but it's probably more like 10-20% here in the upper Midwest. And so we have all these other strains that obviously are infecting people, because Minnesota and Wisconsin also a highly endemic area. But if you look at the states, they rank like number 9 and 11 or so in terms of reported Lyme disease cases. Some of it might have to do with this relationship that as you have more overlap, like on the right side of this curve, you have less of this Borrelia burgdorferi RST1 type strain, which is the more longer lived, more quote unquote virulent strain. So yeah, so that might have bearing on those. So Rick Osfeld was part of this group, Levy et al. They found that with lots of, they have great data over time dragged from their site in the Northeast, which is actually, it's southeastern New York there. And what they found was that, and you can just look at the bottom cartoon, is that they did find that nymphs and larvae, their phenologies, they started becoming active sooner in the year, all right, because it was warmer. However, it seems like the distance or the timing, between the nymphs and larvae actually didn't change. So potentially that, then, there might not be as much of an effect then on phenology in this case, on transmission, unless that also has some interaction with the hosts. But my feeling, I know from speaking with mammalogists that Paramyscus leucopus, which is the main reservoir in many of our areas here for the black-legged, for Borrelia burgdorferi, its biology is such that it takes advantage of when it gets warmer, and it comes out and it starts having babies. So probably with regard to the Lyme disease cycle here, it won't be a big deal. I just want to end with saying that other factors might, even though we can see how climate change might affect the biology and therefore survivorship and maybe even pathogen transmission cycles, other factors might just be, more important. And so we know that also climate change, might affect vegetative communities, host communities. Those will be on a different time scale potentially. And also ticks may disperse to places because they're doing well with the warmer climates or something. But if they get to a place and there are no hosts, or they're the wrong hosts, then they're not really going to spread. And so, for instance, we get tropical ticks that are brought up by birds all the time. Perhaps there'll be more and more with time, but if they are generalist ticks, maybe they would do okay in our areas. But if they're not generalist ticks, then even though they're being imported, they may not establish a population. And then within the ticks, even though we talk about Ixodes scapularis as one species, it seems like subpopulations may have different genetics. And so then the role between genetics and plasticity, meaning and their ability to evolve, might be important. And this is important to consider because when people model the spread of any species due to climate change or something else using niche modeling, if they base their niche on, just one standard type of the tick, it will, it'll produce the projections that are produced are only as good as, I guess, how representative that model tick they used. But for instance, in the north and the south of Ixodes scapularis, I'll just show that one. They're very different, and this is Isis Arsenault's work. But in the north, the ticks really feed on the mice, and in the south, they really feed on lizards. We don't know if that's host preference. We don't know if it's host-seeking behavior, because we know that the ticks in the south rarely come above the leaf litter to host seek. They're mostly down below. So as northern ticks move south, are they going to increase the amount of Lyme disease in the south because they're going to be host seeking above, feeding on mice and all that? Or are they going to revert to the lizard lifestyle because more lizards are around than mice? So that's, I think, a easy type of example to be thinking about. But our student Jenna and then others, they looked at how there are differences in the actual response, I guess, to temperature and for development and such. And there are different responses too amongst the populations. And so how the interactions between genetics and climate work out may also affect our predictions. So I think with that, I'll just stop. And that's about it. Thanks for staying over to 6:06. If anyone wants to stay for questions, I'm here as long as you want, but I don't expect that, I mean, if you have to leave, that's fine.
So I have a question.
Yes.
I was wondering, I understand you work mostly with Ixodes scapularis or only with Ixodes scapularis, but do you find Lone Star ticks up there or American dog ticks as well?
We certainly find plenty of dog ticks, and the Lone Star ticks we're just getting. But that's exactly a perfect example of those ticks that they had been showing up to Michigan like since the 80s, probably dropped off by birds. There are records of those, actually higher number than Ixodes scapularis back then. But they don't do so well in colder climates. And so, in the Northeast, they've been spreading north. I know in Wisconsin, they've been spreading there. Michigan, we're just starting to get them. And I think that's all due to the warming climate, because certainly we have the hosts.
Because what I'm curious about is so we have all three species here, but certainly Ixodes is the least frequent that I run into, at least with flagging and such.
Yep.
However, if I go to a check station in November, the only thing I'm finding on deer that are brought in are Ixodes scapularis. So to me, I was thinking it'd be interesting to do a competition study with the different species. Has something like that been done?
Not in a very manipulative way. So we do know that there's a study you might want to look at by Robert Jordan and Terry Schultze. Now, that study, I think it was published in the 90s, maybe even early 2000s, actually. But, they have been looking at black-legged ticks and then the influx of Lone Star ticks in New Jersey. And supposedly, according to them at that time, they did not see any decline in Ixodes scapularis when Lone Star ticks increased. I don't know if that has changed over time, but I think because they both use deer a lot, I think it's reasonable to think that there could be competition. Not necessarily physically, well, in the spring they could be physically on the host together, but also maybe mediated by salivary proteins and immunity.
Great, thank you.
But if you want to do that, go for it. It would be awesome, amazing, yeah. But yeah, you should try to somehow look at deer outside the hunting season.
Yeah, that's a bit of a problem. I've been trying to look at deer
Yeah.
and turkeys. Mostly
Right.
what I study is ehrlichiosis, because that
Right.
is in 25% of all ticks that I found here.
Ugh.
Yeah.
Yeah.
But in Lone Stars are 99% of what I
Yeah.
capture.
Right.
But dermacentor are carrying ehrlichia as well. There's so many questions still with ticks. It's amazing.
There are. And I'm glad that, you know, other people are studying these other very important ticks. So.
Other questions for Dr. Tsao?
Yeah, Dr. Tsao, this is Bineke.
Hi, Bineke.
I called in late. I'm sorry. I realized halfway through that your talk was going on. But I wondered, now that, so when we're talking about climate change, and if you think about the different sort of, well, genetic variability in the different inner ticks, we still do a horrible job at predicting if it's going to be a good or a bad tick here.
Yeah.
Do you feel like we can be better at that with, like, if you think about the growing decrease days and those pieces?
Well, I think part of it is that we could probably predict very well longer term trends, but, you know, just like when people are interested in looking at how trophic cascades, you know, those are, or like with the acorn masting and such, should increase ticks, like, you know, not densities of infected ticks in 2 years. What happens is that if you get a very dry year, that even if the biotic processes were happening that way as predicted, if you have a dry year, all of a sudden it will just wipe out the ticks, you know? And so we might be able to model then, I guess, the variability in, but I don't know, you maybe be able to say whether it'll be good or bad year, you know, will be more variable than in the past. But if we get better, I guess, at predicting when these rains might come to make, or cooling episodes, I guess, because of the polar vortex or something, maybe we'd be able to predict a little bit more, but I'm not sure. Not me, but maybe you could do it.
I find it such a hard question. It's like, yeah, we had a lot of freeze thaw. So in your area, maybe like they died. I'm not sure.
What did you say? What happened?
Yeah. So when people ask us here in Wisconsin, so I'm located in Wisconsin.
Right.
And you think about like our nymphal and adult populations, it's like, okay, they're fine if they're under snow cover. But when we have a ton of freeze thaw,
right,
which we have in certain locations, and especially the last couple of years, you're like, I don't know what to tell you. And people have no doubt. That's like the first question they often ask us, right? When we ring the doorbell, it's like, is it a bad year?
Right.
I don't know.
So hopefully people should do like what you're talking about and take data on that. We should take data more on these freeze thaw events, because certainly that would affect the ticks. You know, the other thing is, the other thing is, it depends a little bit on the host population, right? Because what happens is if there's a lot of hosts during a certain year or certain time, they are competing with us, you know, in terms of picking up the ticks. And so unless you're trapping and dragging, you know, and maybe seeing what get on people, it's, you're not seeing the whole picture.
Agreed.
Yeah, I think
we can keep working on it.
Yeah, I think a few years ago, it was when it was, I can't remember which year it was, but in the Northeast and here, many of us had a hard time finding nymphs. With something like it was too dry, too hot. I think that's what it was. And we could not drag nymphs, but yet the fall adult peak was fine. So obviously the ticks, either through behavior, you know, of just really digging in low, and like I said before, maybe they're getting on host just fine in that way. And I know your lab, you know, has tried to study whether if it's hot, you know, will they host seek more at nighttime? And I have not done that around here, but down in Florida, that didn't seem to be the case. But I think you guys may have found something that they may have a little bit more. So again, we're not dragging except under experimental conditions, you know, at nighttime. And so maybe the ticks are getting on the host then, switching their behavior. And that's why we saw a lot of adults.
That's fascinating. Thank you for your talk.
Yeah. We'll have to thank Christy again for having me.
Oh, absolutely. It's my pleasure. And this was fascinating. I think it was just this morning, the undergrad who gets to present his honors thesis, just through a little bit of data instead of everything we wanted, was showing us his data. And we were asking questions that you have nicely given us some answers to. So it was
good.
Yeah, to get some more thoughts on this.
Right.
Any last questions from the audience?
I got one. This is Ryan Larson, also from the Paskowitz Lab at the University of Wisconsin.
Yes. Hi, Ryan. Right.
As I think Sarah Hamer pointed out, there's a synchronous phenology that we see in the upper Midwest, especially the northern parts. And just something I had in the back of my mind throughout your presentation is, if that were to shift to an asynchronous like we see in the Northeast, could that actually increase possibly could it be a downstream effect where it possibly increases the density of nymphs the following year because mice populations increase throughout the summer, so there's way more hosts in August versus June that you would see with the typical synchronous larval peak?
Yeah, I think that's a great question. And for me, I, in part, wonder why is it, like when I talk about the diapause, I thought, why is it that some of these ticks don't host-seek more at the end of summer? Because they're way more, the biomass of hosts is much greater.
There's like 2 mice out in May, and it's frustrating if you,
right.
trap in May.
Yeah, right. But I think that's why the peak is more like June, July. However, it must be, and there is some data about it, that the survivorship of engorged, I guess, larvae, perhaps, or maybe it's just not as good, or that they actually, maybe they survive just fine, because I think they say they survive, but maybe they're at a disadvantage the next year when they're host seeking. But if you look at the Duke-Wasser et al. Study where they we dragged for ticks in the Northeast and Midwest, North and South, the densities of ticks overall, there was variation in the North and in the Northeast, as well as variation in the Midwest. And so maybe we didn't look carefully enough, but I think the overall thought was that the densities were pretty much the same. But you could be right that, if we had more of the asynchronous, maybe the success rate of those larvae feeding could be higher. But then I guess we'd have to see with their, molting success and such for the next year.
Thank you.
Yeah. And I'll send you guys in Wisconsin some other data that we've had.
Oh, that'd be great.
Interesting. Yeah.
Thank you.
All right. Is it time for beer or wine?
Yeah, I think it's happy hour time.
Our second week of, COVID-19, kind of isolation. So everyone's having to deal with that. So we deserve it.
Absolutely. So everybody either find a Zoom friend or have a beverage with somebody in your own household. But again, thank you so much. This was fantastic. And to use the really awful meme that's going around, what goes better with Corona than Lyme?
That's right. Actually, I hadn't seen that, so thank you. And I hope you have a very good trip out east. I assume you're going, or are you not? Or
yeah, so we actually got to set up our fields last week. In the first couple days before our Iowa governor, told us to come home. So our grids are set up, and we are using the little devices that they used at the field station in Connecticut. So,
good.
Is now being distributed. We hope to, actually begin trapping creatures, maybe end of April. But at the very least, we have permission to go back out and refill those stations. So the trial has started.
All right. Very exciting.
Yeah. Thank you.
Yeah. So, all right. Well, good luck, everyone. And surviving, and hope your work's not impacted as much. And, and I hope to meet you all. In person one day.
Absolutely.
Yeah, and anyone's, welcome to write me if they have other questions or about anything. Yeah.
So it looks like the chat is blowing up, thanking you. So
I appreciate it. Yeah. All right. Take care.
yep, you
Bye-bye.
too. Bye.
Bye-bye.
Bye bye.
Ciao, ciao.
Bye.